you can anytime book a slot with us for a personalized counseling. We have 3 different subscription plans, starting from basic, which is for 15K, going up to 50K. That is our premium plan for 12 months. And you can avail extra 20% off by using my promo code as displayed on the screen here. So our next topics will be that we'll be talking about is cholesterol metabolism and lipoprotein metabolism. Okay, so let's begin our today's topic, which is fatty acid synthesis. So beginning with the introduction as usual, that when does fatty acid synthesis takes place in the body? So it takes place in the well-fed state under the influence of insulin. So insulin is an anabolic hormone and your synthesis cycle is an anabolic process. So your fatty acid synthesis is taking place in the well-fed state under the effect of insulin. A fatty acid is synthesized in the body and is stored as TAG in adipose tissue. TAG is your triacyl glycerol. So your fatty acid is majorly stored in adipose tissue. That is, your fatty acid combines with glycerol to form TAG. That is triacyl glycerol. Palmitic acid is the most common fatty acid synthesized in the body. And the site of fatty acid synthesis is liver, adipose tissue, kidney, brain, mammary gland. And the organelle where it is taking place is it is extra mitochondrial, that is cytoplasmic cycle. And it is also known as Lenin spiral. This is based on the scientist name which elucidated the maximum steps in fatty acid synthesis. Now, this pathway needs energy. Okay, so your fatty acid synthesis requires energy. The starting material is acetyl-CoA. Now, where is this acetyl-CoA coming from? The acetyl-CoA is coming from the link reaction and beta oxidation of fatty acid. Now, both of which takes place in the mitochondria, as we already know. So, acetyl-CoA has to be transported to the cytoplasm for fatty acid synthesis. Now, your acetyl-CoA link reaction, you know what is it? Aerobic glycolysis. Where your pyruvate that was formed from glucose via glycolysis converts into acetyl-CoA, and the enzyme was pyruvate dehydrogenase. Okay, so these are the sources of your fatty acid synthesis. Initial substrate, that is starting material, acetyl-CoA. So what is the source for acetyl-CoA? Aerobic glycolysis, the link reaction, and your beta oxidation of fatty acid. Now, what is it saying? That this acetyl-CoA is formed in the mitochondria, but the fatty acid synthesis is taking place in the cytoplasm. So, the initial starting of fatty acid synthesis will be that first you have to transport that acetyl-CoA from the mitochondria to the cytoplasm. Okay, so now the basic steps of fatty acid synthesis, what are they? The first step is transfer of acetyl-CoA from the mitochondria to the cytoplasm. The second is acetyl-CoA carboxylase reactions, and the third is fatty acid synthase complex reactions. Now we are going to take each one of those steps in detail one by one. So this transfer of acetyl-CoA from mitochondria to the cytoplasm. This is your mitochondrial matrix, and here is your acetyl-CoA. So this acetyl-CoA, I'll zoom in. I hope it's visible now, which is 2 carbon, has to be transported into your cytosol here. Okay. Now let's see how this is happening. So this 2 carbon acetyl-CoA, this combines with a 4 carbon oxaloacetate The enzyme that is acting is citrate synthase to form citrate. And what is citrate? Citrate is a 6-carbon tricarboxylic acid. And this tricarboxylic acid, it has a transporter known as tricarboxylic acid transporter in the inner mitochondrial membrane. Okay, so this citrate travels via that transporter into the cytosol. Easy. Your 2-carbon acetyl-CoA combines with 4-carbon oxaloacetate to form 6-carbon citrate. This citrate is a tricarboxylic acid, and it has a tricarboxylic acid transporter for it present in the inner mitochondrial membrane. The outer mitochondrial membrane is almost permeable to all the compounds, including acetyl-CoA. So this citrate, it travels here. So the inner mitochondrial membrane is bypassed via this transporter, and now this citrate reaches into the cytosol. What happens in the cytosol? That it is acted upon by ATP citrate lyase. So an ATP molecule is used, and this citrate is split into again acetyl-CoA and oxaloacetate. Now you see your acetyl-CoA has been transported in the cytosol. So we are done with the first step, that is transfer of acetyl-CoA from mitochondria to cytoplasm. Moving on to the next step, which is acetyl-CoA carboxylase reactions. Now let's see what is happening in this. Okay, so you can see your acetyl-CoA here, acetyl-CoA. This acetyl-CoA is 2-carbon, as we know, and we can also see that. And you can see here on this side what is being formed, malonyl CoA. So your malonyl CoA is 1, 2, 3 carbons. Okay, so you can see a 2-carbon compound is changing into a 3-carbon compound. So this is a carboxylation reaction. A carboxylase enzyme is being used, and this is a carboxylation reaction where a 2-carbon compound changes into a 3-carbon compound. So a carbon dioxide molecule is needed. So where is this carbon dioxide coming from? Bicarbonate donates this carbon dioxide. So this carbon dioxide is donated by a bicarbonate ion. So we can see your 2-carbon acetyl-CoA changes into 3-carbon malonyl CoA. A carbon dioxide molecule you can see is being added, and as we know, all carboxylation reaction, all carboxylase enzyme need biotin and they require ATP. So biotin and ATP are required, and your acetyl-CoA via the action of acetyl-CoA carboxylase enzyme changes into malonyl CoA, which is a 3-carbon compound. So that was the second step of fatty acid synthesis. That was the reaction of acetyl-CoA carboxylase. Now moving on to fatty acid synthase complex reactions. Here. So the reactions of fatty acid synthase complex, it is the longest phase in fatty acid synthesis. Okay, so this is the longest phase, and the most common fatty acid synthesized in our body is palmitic acid, and all other long chain fatty acid and very long chain fatty acid, they are elongated on this palmitic acid. Okay, now before studying the reactions of fatty acid synthase, let us see what is this fatty acid synthase complex. And from here, if you don't uh, follow me very like precisely. If you miss anything, it will not be clear to you because it'll get a little complicated, huh? So please listen very carefully because these steps are a little complicated. So, and to understand the reactions of fatty acid synthase complex, it is very important that you understand the complex, the structure of the complex. So please hear very carefully without missing on any points. And I'll go slow and I'll repeat it. Okay, so what is fatty acid synthase complex? You get asked about this in exams a lot. You can get it in short notes for 5 marks. Write about fatty acid synthase complex, just the complex. Okay, so you need to know this. Now, what it is a homodimer. What is a homodimer? That is two polypeptidanes which are similar. So it is made up of two monomeric units which are similar. So it is a homodimer. You can see here in this, the primary structure has been shown. Okay, so two similar such chains, they form a homodimer. And how are they arranged? The X-ray crystallography has revealed that it has a X-shaped. Okay, so it is X-shaped. The two of the monomers are arranged in such a pattern that they form an X like this. Okay, this is monomer 1, this is monomer 2. You can also see in the diagram that it is arranged in the form of an X, and this was revealed by X-ray crystallography. What is X-ray crystallography? It is a technique used to study 3D or tertiary structures of proteins. Okay. Now, each of the monomer unit has 6 enzyme activity and one acyl carrier protein ACP. Okay, you can see different enzyme activities here, 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 here. So each of the monomer has 6 different enzyme activities and one ACP. What is ACP? Acyl carrier protein. ACP has a pantothenic acid in the form of 4-phosphopantothenium. Okay, if you don't even remember this, then at least you should know that it has a pantothenic acid group. You should at least be knowing this. The Fas complex is a multifunctional enzyme. That is, a single polypeptide chain has more than 2 enzyme activity. We have studied about bifunctional enzymes. I hope you remember where we have studied bifunctional enzymes. If not, then you need to revise. We have studied bifunctional enzymes twice, and I have told you there that what is a bifunctional enzyme? Where a single polypeptide chain was having 2 enzyme activity. In glycogen synthesis and in gluconeogenesis, we have studied this, okay? So what is Fas? Fas is not a bifunctional enzyme. It is a multifunctional enzyme. And what is a multifunctional enzyme? When the polypeptide chain is having more than 2 enzyme activity, okay? So we have already said that it has 6 enzyme activity, okay? 6 different enzyme activities are there. So that was briefly about Fas, okay? I hope it is clear that it is an X-ray, X-shaped structure, and it is a homodimer, and each monomer has 6 different enzyme activities plus one ACP, that is acyl carrier protein. So this was about Fas, and Fas has is a multifunctional enzyme. Obviously, when we are saying it has 6 different enzyme activities, so it is a multifunctional enzyme. Okay, now each monomer has 3 domains. So it has been categorized into different domains. The first domain, second domain, and the third domain. The first domain is the condensing unit. The second domain is the reduction unit. And the third domain is releasing unit. So that is how it has been categorized. Now, the first domain has 2 enzyme activity. That is acetyl malonyl transacylase and ketoacyl synthase. Now, in older editions of Harper, they were mentioned as 2 different enzyme activities. This acetyl malonyl transacetylase was mentioned as 2 different enzyme activities, but in newer editions, you will find that they have been mentioned as only one enzyme. The second enzyme activity of the first domain is ketoacyl synthase. Now your second domain is the reduction domain, and it has three enzyme activities, that is ketoacyl reductase, dehydratase, and enoyl reductase. And the third domain only has one enzyme activity, which is thioesterase, and it also has ACP, that is acyl carrier protein. Here in the diagram, you can also see how it has been divided into different domains. This is your first domain. This is your second domain. And this is your third domain. Okay. And each domain has different enzyme activity. So that was about FAS. Now moving on to the reactions that are carried out by FAS. For the sake of convenience, let's assume that we give the name to the monomers as monomer 1 and monomer 2. Okay. Let's this one be monomer 1 and monomer 2. I'm just giving them the name so that it's easy for me to explain and it is easy for you to understand. So there is a cysteine group in the monomer 1. Cysteine we know that has a sulfhydryl group. Okay, and in the monomer 2, the ACP of monomer 2 that also has a sulfhydryl group. This is exactly what the diagram is showing here. You can see this is your FAS. So the first monomer has a cysteine group which has SH group, sulfhydryl group, and the other monomers ACP has this SH group. Okay, now let's see what is happening. So an acetyl CoA, this acetyl CoA from acetyl CoA, this attaches to your, first it will attach to ACP and then from ACP it shifts to cysteine. Okay, some books they will skip this step and then directly tell you that the acetyl from acetyl CoA attaches to the cysteine SH of monomer 1. So this is easy for you to remember. So you can also skip this step because first it attaches to ACP sulfhydryl group and then it shifts to cysteine. So this can also be omitted. Okay, such great details are not required at your level. So your acetyl from acetyl CoA attaches to the SH of cysteine in monomer 1. Okay, acetyl group attached here, 2 carbon. And your malonyl here from malonyl CoA attaches to where? The ACP sulfhydryl group. You can see this. So here in my hand diagram, what is attaching here? Malonyl. And your malonyl group is 3 carbon. Okay. And this is brought about by the first enzyme, that is acetyl malonyl transacylase enzyme. Okay. So the enzyme is acetyl malonyl transacylase. Okay, I hope this step is clear. I'll repeat it once again. See, 2 monomers, monomer 1, monomer 2. Okay, the monomer 1 is having a cysteine group with sulfhydryl. And this ACP also has a sulfhydryl group. Now your acetyl group attaches to the cysteine and your malonyl group attaches to the sulfhydryl group of ACP. And this is brought about by the enzyme acetyl malonyl transacylase. Okay, that was simple. Now let's see what is happening. Decarboxylation is taking place in the next step. We have already seen how your malonyl was formed. Your malonyl was formed by addition of this carbon group. Carbon dioxide was added. Okay, now this carbon dioxide which was added by your acetyl CoA carboxylase is being removed. Okay, it is liberated. And now your acetyl, which was there on the cysteine first monomer, it condenses with your malonyl on ACP. See here. So your acetyl condenses with malonyl on ACP. Okay. Now what you see, what has been formed because of this? A 3-keto acyl group has been formed. This is a 3-keto acyl group. You can see there is a keto here. Okay. So this is a 3-keto acyl group. Now the next 3 reactions, that is this and this. These 3 reactions that will take place, this will take place to produce a corresponding saturated acyl group. Okay. To saturate this acyl group. And this will be just the reversal of beta oxidation. Remember, in beta oxidation, I told you the covalent bond is very small. So we need to weaken the bond. And what were we doing? We were first introducing a double bond, then we introduced a hydroxyl group, and then we introduced a keto group. And then when the bond energy shifted, lysis occurred. So what is happening here? This is just the reversal happening. You can see there is a keto group. Now to the keto group, we will reduce it to a alcohol, that is a hydroxyl group will come, and then we will introduce a, you can see, a double bond, and then that double bond will be changed into a single bond. Okay, so the next 3 steps are just happening to reduce that keto into a saturated acyl group. Okay, let's see how is that happening. And for this decarboxylation step, the enzyme was acid ketoacyl synthase, which is the condensing enzyme. Okay. Ketoacyl synthase. See here. Now in the next step, what is happening? That your keto is being reduced. This keto group is reduced to an alcohol. You can see here. So this keto group, alcohol, and the enzyme is ketoacyl reductase. And in this reaction, NAD plus is being produced. Now what happens next? A water molecule is removed. Okay, a water molecule is removed to create a double bond between alpha and beta. You can see a water molecule is being removed. So this water is removed to create a double bond between alpha and beta carbons. You can see a double bond has been formed here. Okay. And the enzyme here is hydroxyacyl dehydrogenase. Okay, so enzyme used here is hydroxyacyl dehydrogenase, as it is removing a water. Okay, now this double bond has been introduced. Now this double bond is reduced to a single bond. You can see here. This double bond is reduced to a single bond. So your double bond reduced. And this is brought about by the enzyme enol reductase. And you can see what has been formed here. A 4-carbon saturated fatty acyl, okay, which is present at the ACP position. This is a little complex. I'll quickly go through it once again, okay. Okay, so these are your 2 monomer units. Monomer 1 which has a cysteine group and the monomer 2 which has an ACP. So both of them have sulfhydryl groups. Okay. Now what happens is that your acetyl from the acetyl CoA is transferred to the cysteine, and your malonyl from malonyl CoA is transferred to your ACP. The enzyme is acetyl malonyl transacylase. Okay, you can see this is what is formed. An acetyl at the cysteine position in one monomer and a malonyl at ACP in the second monomer. Now what happens that how was your malonyl formed if you remember? It was formed by the action of acetyl-CoA carboxylase. So that carbon dioxide which was added is removed here. Okay, so decarboxylation is taking place and this is brought about by the enzyme ketoacyl synthase, and what is being formed? 3-ketoacyl group is being formed. You can see a keto group here at this position, at the third position. Okay, so what has been formed? A ketoacyl group has been formed which needs to be changed into its corresponding saturated acyl group. Okay. Now, how is that taking place? Now, the next reactions are to convert that keto group into its corresponding saturated acyl group. So, first, that keto group is reduced to an hydroxyl. Then, that hydroxyl, by removing water, you introduce a double bond between alpha and beta carbon. Then, that double bond is reduced to a single bond here. And then you can see a 4-carbon saturated fatty acyl group has been achieved, okay, which is at ACP position. The enzymes, you can see the names. This is ketoacyl reductase. The enzyme used here was hydroxyacyl dehydratase, and used here was enolide reductase. Even if you don't remember the proper names, you know that your keto group is being reduced to your hydroxyl group, so simply write reductase. Okay, now you know that you're removing water, so write dehydratase. Now you know you're reducing a double bond to a single bond, so simply write reductase. Okay, the name of the enzymes, if you know the function, so it's okay. Even if you simply write reductase and dehydratase, it'll serve the purpose. Okay. Similarly, for this enzyme, ketoacyl synthase is very simple because you know you're producing a ketoacyl group here at this position. Once again, I'll take this briefly, okay, so that you learn it while I'm explaining it to you. Two monomers with cysteine and ACP. Now your acetyl attaches to the cysteine and your malonyl attaches to the ACP of two different monomers. Then what happens? Decarboxylation takes place. Your carbon dioxide is liberated and your acetyl from the cysteine condenses with the, you can see here, your acetyl from the cysteine condenses with the malonyl at ACP to form a 3-keto-acyl group. Now, the next three reactions are just to convert that keto into saturated acyl. And what you get is a saturated acyl at position ACP. Now you see what is happening. Now this butyryl ACP goes to the cysteine, okay, of the monomer 1. So this is a repeat step. Now you can see another malonyl comes and attaches to the ACP. Then again decarboxylation takes place, and then this butyryl group condenses with the malonyl at ACP. So you can see what is formed: 1, 2, 3, 4, 5, 6. A 6-carbon ketoacyl has been formed. Okay. Now again the next three reactions will take place to produce the corresponding saturated acyl group. So you can see the corresponding 6-carbon saturated acyl ACP has been produced, which is known as hexanoyl ACP. Then again this will repeat. This will repeat number of times to produce a palmitate. Okay. So that is how your fatty acid has been synthesized. So that was about fatty acid synthesis. I hope it's clear. I've repeated it many number of times so that it's clear. It's a little bit complicated, but I hope you have understood how it is happening. So moving forward, the regulation. So the regulation is of two types. Short-term regulation, which is very short acting and fast. It consists of allosteric regulation, covalent modification, and compartmentalization. And then you have long-term regulation. If the body has enough acyl CoA, that is fatty acid, it will decrease the expression of enzymes that synthesize fatty acid. Okay. Now let's see what is allosteric regulation. जो acyl CoA carboxylase is the rate limiting enzyme. So the rate limiting enzyme of fatty acid synthesis is your acyl CoA carboxylase. And your acyl CoA carboxylase is favored by citrate. So citrate is enhancing it, and it is inhibited by long chain fatty acid. Citrate is also an activator of TCA transporter, that is tricarboxylic acid transporter. And long chain fatty acid is also an inhibitor of TCA transporter. So you can see this is your tricarboxylic acid transporter. It is being activated by citrate and it is being inhibited by long chain fatty acid. Here in this case, it is palmitoyl CoA. So it is inhibiting this. And citrate activates this transporter. So also you can see that your acyl CoA carboxylase is inactive in its dimer form and it is active in its polymer form. So when these dimers polymerizes, they become active. And how these dimers polymerizes? They are polymerized by citrate. So citrate favors your acyl CoA carboxylase by polymerizing the inactive dimers. Okay, and it is inhibited by your long chain fatty acids. Okay, so your acyl CoA carboxylase is inhibited by long chain fatty acid. That is it. What will they do? They will turn the active polymer into inactive dimer form. And on the other hand, your citrate, what they do is they turn the inactive dimer into polymer form. And also citrate activates the TCA transporter. The TCA transporter, as we have seen, it was necessary because it was involved in transporting of acetyl CoA into cytoplasm. Okay, so if that transporter is blocked, your acetyl CoA will not be transported in the cytoplasm. Fatty acid synthesis will not take place. And your long chain fatty acid, that is when there are already enough fatty acid being synthesized, so that transporter is inhibited. Okay, so further fatty acid synthesis will not take place. So that was about allosteric regulation. Now covalent modification. Covalent modification is brought about by phosphorylation and dephosphorylation. Covalent modification we have studied in all the pathways. We have studied allosteric modification also in all the pathways. And by now, I think it must be like on your tips that what is insulin doing. Your insulin is the enzyme. We have already seen that fatty acid synthesis takes place in the well-fed state. So what was the hormone of the well-fed state? It was insulin. And what was insulin doing? It was dephosphorylating rate-limiting enzymes. On the other hand, we had also studied that your glucagon, what was it doing? It was phosphorylating the rate limiting enzymes. So if fatty acid synthesis is under the influence of insulin, so we know that acetyl CoA carboxylase, which was the rate limiting enzyme, is going to be active in the dephosphorylated state. Okay. Now let's understand this with the help of a diagram. So acetyl CoA carboxylase, here we can see it is inactive. Why is it inactive? Because we can see it has been phosphorylated and it becomes active when it is dephosphorylated. So this is the dephosphorylated state. How does it attain that dephosphorylated state? When it is acted upon by protein phosphatase, because protein phosphatases, they remove that phosphate group from it, and this protein phosphatase, it is activated by insulin. So that is how your insulin dephosphorylates enzyme. So when insulin is there, protein phosphatase will be active, and when protein phosphatase is active, it will remove this phosphate from the inactive acetyl CoA carboxylase to make it active in its dephosphorylated state. And on the contrary, when glucagon or epinephrine is there, what they do through those cascade of events that we have already studied, they activate kinases and cyclic AMP and everything, and that cycle goes on. And ultimately, in the end of the cascade, we had seen that they had activated these protein kinases, which what they do is phosphorylate enzymes. Okay, so this dephosphorylated, which was active acetyl CoA, will be phosphorylated, you can see, by this kinase, and then it will become inactive. So that is how your glucagon is making acetyl CoA carboxylase inactive. And on the other hand, your insulin, it makes the acetyl CoA carboxylase active. Now the other thing is compartmentalization. So that is the third form of regulation. The beta oxidation we had studied was taking place in the mitochondrial matrix. Fatty acid synthesis, as we have just seen, takes place in the cytoplasm. So both the cycles are taking place in different compartments of the cell. One is taking place inside the mitochondria, the other is taking place in the cytoplasm. So both of the cycles are separated from each other with the help of compartment which is impermeable to each other. So if they were taking, both of them were taking place in the cytoplasm and were not regulated, so on one hand you would be creating fatty acids, on the other hand you will be breaking down. So this compartmentalization is also a way of regulation. Now, elongation of fatty acid. The fatty acid synthesized is only 16 carbon long, okay, because the most common fatty acid being synthesized, we have studied, I told you, is palmitic acid. So it is only 16 carbon long, but the body needs longer fatty acids, especially your brain, myelin, okay. So therefore, elongation of fatty acid needs to take place. Now, elongation occurs in smooth endoplasmic reticulum by an enzyme complex called microsomal fatty acid elongase system and mitochondrial fatty acid elongase. So these two enzyme systems are involved in the elongation of fatty acids. So this is the major site. The elongation is taking place, and this is the minor site. Especially during the myelination of the brain, your elongation process is increased. Okay. A synthesis of unsaturated fatty acid. This takes place with the help of two enzymes, elongase and desaturase. Okay, so they remove the hydrogen bond and make saturated fatty acid into unsaturated. So your desaturase, they remove hydrogen and make saturated fatty acid into unsaturated. Okay. And the site is endoplasmic reticulum. So where is your saturation or unsaturation of fatty acid taking place? It is taking place in the endoplasmic reticulum with the help of two enzyme systems, elongase and desaturase. Your desaturase, it removes the hydrogen and makes the saturated fatty acid into unsaturated fatty acid. And your elongation was taking place in smooth endoplasmic reticulum and mitochondria fatty acid elongase system, and your smooth endoplasmic reticulum was the major site, whereas the mitochondria was the minor site for elongation. And this elongation process is increased during myelination of brain, because your brain, especially the myelin, it needs longer fatty acids. Okay. So that was about fatty acid synthesis. I hope it's clear, especially these steps because they were a little complicated. Okay, so that was all about fatty acid synthesis. Thanks for watching. If you have any queries, you can ask. So with that, we come to the end of the session. Thanks for watching the live. In the next class, we'll be talking about cholesterol synthesis.